Hello and welcome to another Bio43 pre-lab video. This should be the second video you watch before the first week of lab. One vital thing to do at the start of a course is look over the syllabus. There's a lot of important policy information you should be aware of here. But I want to take a moment and focus on the learning objectives. As new scientists, these are the things you should be able to do at the end of this course. These are actually the learning objectives from the fall 2019 syllabus, the last time Bio43 was taught in person. I want to show this to indicate how little has really changed in our goals. Even before this move to distance learning, we were less focused on developing your physical skills to perform research than your ability to think critically about experiments. Each video, each class, and each assignment will have specific goals that directly relate back to these learning outcomes, which we'll point out in each future video. For instance, in the first video, understanding the types of journal articles provides the foundation for applying knowledge from the scientific literature to an experimental context, and for identifying underexplored questions of the natural world. This video discusses hypotheses and predictions and provides an example of how to design an experiment. It will be an important reference for you when it's time for you to construct testable hypotheses and thoughtfully design experiments. Let's start with a hypothesis, a proposed explanation for an observed phenomenon. For instance, say you've observed trees losing their leaves during the winter, and you want to know why. Pause here for a moment to think of a few ideas. Maybe you thought it's due to shorter days, or lower temperatures, or changes in weather patterns, or any of a number of other options. When it comes to unexplained phenomena, there will often be many explanations you can think of, and probably a few you can't. So you rely on knowledge from past observations, experiments, and the scientific literature to decide on the explanation you think is most likely. I'll decide here, what with this being a pre-recorded video, but in class, you'll come up with the ideas in groups, and then vote on which hypothesis will be investigated. In this case, let's say we decide shorter days is the explanation we want to investigate. Our hypothesis is trees lose leaves in winter due to decreased daylight. I want to emphasize how a hypothesis differs from a prediction, since this is often a point of confusion. A prediction is what you think will happen in a specific experiment if your hypothesis is correct. At this point, we can't even say what our prediction is, because we haven't designed our experiment. A good hypothesis could be tested by multiple experiments, each with a unique prediction. Think of a hypothesis as a more general statement of how nature works, typically written in the present tense because it describes a phenomenon occurring in the world right now. In contrast, a prediction is a specific prophecy about the results of a given experiment, typically written in the future tense. It only describes what will happen in an experiment you're about to perform. Now let's design an experiment to test our hypothesis. We proposed a causal relationship. Decreased daylight causes leaves to fall. So we can identify the independent variable, the variable we alter in our experiment, as the amount of daylight trees receive, and the dependent variable, the variable we measure, as the number of leaves that drop in a given time frame. It is important to classify these variables as categorical, consisting of non-numerical groupings, discrete, consisting of indivisible units, or continuous, theoretically divisible into an infinite number of pieces. While these terms are simple, there are some tricky practical applications. For instance, the dependent variable here, number of leaves, is discrete. A tree doesn't drop a fraction of a leaf. But when we look at the average number of leaves that fall from a group of trees, we do get decimals, so we treat this as continuous. Meanwhile, our independent variable, amount of daylight, is continuous, but we can simplify things by treating it as categorical. Rather than trying to measure a wide range of values, we can just measure one condition where the amount of daylight is constant and one where it varies. Pause and take a moment to think of how you would design an experiment using these variables. Here's one way to set it up. In our control treatment, we'll use light fixtures to expose a group of trees to 12 hours of light, followed by 12 hours of darkness, consistently for 180 days. For our experimental treatment, we'll take other trees and start by exposing them to the same 12-hour light, 12-hour dark cycle, but gradually reduce the amount of light each day over the same period of time until they have 8 hours of light and 16 hours of darkness. We'll count the number of leaves that fall from each tree over the 180 days of the experiment. Ideally, the independent variable should be the only thing that differs between the two treatments. We want to control everything else as best we can. Make sure the trees in each group are of the same species, same age, same state of health. Make sure the sites are close enough together that they experience the same temperatures and weather patterns. Take a moment and think about other variables you would want to control in this experiment. However, not everything can be controlled for. Such factors are called confounding variables. Maybe one treatment site ends up with more cloud cover, or more nutrient-poor soil, or more leaf-eating herbivores around. These things could change the measurements for the dependent variable, and if they affect one treatment differently from another, your resulting conclusion could be unreliable. How could you know that a difference between your treatment groups is due to your independent variable and not due to a particularly impactful confounding variable? It is also important to consider our biases. We want our hypotheses to accurately describe nature. As scientists, we value honesty and integrity, so the vast majority of us would not falsify data on purpose. But subconscious biases can affect our results. Suppose some leaves blend in really well with the forest floor and can be hard to notice. Maybe when we collect leaves from the experimental treatment, we're very careful not to miss any. But for the control treatment, we're less vigilant and wind up missing some leaves. This could seriously affect our conclusions. We need to think ahead about the ways subconscious bias could manifest and work to eliminate them. One great way is through blinding. If the person who counts the leaves doesn't know which trees are in the control treatment and which are in the experimental treatment, they can't be biased in their counts. Now we have to decide on the sample size. That would be the number of trees in each treatment, not the total number of trees used. In this case, our sample size will be 8. This means 8 trees in the control treatment and 8 in the experimental. Across our treatment groups, we will count a total of 16 trees, but our sample size is 8 for each treatment. Larger sample sizes are better, and we'll see why that is when we discuss statistics. However, what counts as a reasonably large number varies based on the situation. In some experiments, it's easy to collect hundreds of samples. But counting every leaf that drops from hundreds of trees would be impractical, so a sample size of 8 seems like a reasonable choice here. Trade-offs of experimenter practicality versus scientific rigor are decisions you, the PI, will routinely have to make. Now that we've fleshed out our experiment, we can finally formulate our prediction. The trees in our experimental plot will drop more leaves on average than the trees in the control plot. This is a logical prediction to make if our hypothesis accurately describes nature. So if the results match our prediction, this is support for our hypothesis. That does it for this primer on experimental design. Be sure to check in on this video along with your experimental design and statistics guides when it's time for you to design your own experiments. See you next time.